Hello everyone, welcome to Patho Excellence. You all know skin is the largest organ of our body, and in histopathology section the smallest biopsy we are getting is from skin only. So basically they are often considered difficult to diagnose because of limited tissue, partial representation and overlapping features. But in this series skin biopsy interpretation I will make it easy. So by first mastering the normal histology of skin and clearly understanding dermatology terms used for lesions, you will be able to approach even a tiny biopsy in a systematic and confident manner. So in this video, the first step we will see is uh, mainly about normal skin histology and lesion terminology. So here the outline of this topic is, first of all we will learn dermatology terms, which is the description of skin lesions. When the skin biopsy is received, we are getting some clinical history and the description of skin lesions patient had. Now second topic we will discuss is normal skin histology, and in last biopsy types and processing. So let's start with the uh, skin biopsy interpretation, in which first is dermatology terms. So first is, what is macule? It is a flat lesion. It is less than 1 centimeter in diameter. And what is patch? It is also a flat lesion, but it is greater than 1 centimeter in diameter. So macule less than 1, patch greater than 1. Papule, or mainly it is less than 1 centimeter, solid and raised lesion, while plaque it is also solid and raised but diameter is greater than 1 centimeter. Vesicles are fluid filled lesion less than 0.5 centimeter in diameter, and bulla are same but they are having greater than 0.5 centimeter diameter. So both these terms have been used interchangeably with blisters. And what is nodule? So nodule is a solid, elevated, circumscribed and greater than 1 centimeter size lesion. So these are some dermatology terms which we have to understand when we correlate clinical history and our microscopic findings. So these are some clinical picture of skin lesions like bulla more than 1 centimeter, fluid filled lesion, macule, it is flat discolorated less than 1 centimeter and brown in color, nodule, it is elevated, lesion solid more than 1 centimeter, patch which is more than 1 centimeter, flat and circumscribed discolored, papule which is less than or equal to 0.5 centimeter, solid elevated lesion, plaque it is elevated solid flat but more than 1 centimeter, pustule which is vesicle containing pus, vesicle it is fluid containing lesion less than 1 centimeter in size, will it is edematous, plaque and it lasts for few hours, scale it is epidermal thickening and it is flat or raised, and crust which is dried serum or exudate on skin, fissure which is crack or split, excoriation means linear erosion, erosion means loss of epidermis. So here lichenification, important finding, that is thickening of the epidermis seen with the exaggeration of the normal skin lines, and scar which is thickening and permanent fibrotic change that occurs on the skin followed by damage to the dermis. So these are some terminology we receive when we receive skin biopsy. So let's start with the normal histology of skin. In this, we will discuss epidermis and dermis. So, this is the whole epidermis and dermis part. In this section, we will learn all this terminology one by one. So, what is first of all skin? So, skin is the largest organ in our body and it constitutes around 8 to 20% of body mass, having 1.6 to 1.8 meter square surface area in an adult. It comprising of epidermis, dermis and hypodermis. And what is the function? Mainly protection, thermoregulation and detecting sensory stimuli. Basically, this skin is comprising of different cell types. One is most important keratinocytes, which are nothing but the squamous epithelial cells that originate from the basal stem cells and continuously mature from basal to corneum layer and desquamate. So, it contains keratin, so that is why it is called keratinocytes. Second cell, which is melanocytes, which gives color to the skin and its synthesis melanin and it protects from ultraviolet radiation. Third is Langerhans cells, which is antigen producing cells. Merkel cells are mechanoreceptors. Now epidermis is comprising of 5 layers going from deep to superficial. So basal layer, which is also called stratum basale or germinativum. Prickle cell layer, that is stratum spinosum. Granular layer, stratum granulosum. Clear layer, stratum lucidum and conified layer, it is stratum corneum. Now this prickle cell layer and basal cell layer are collectively referred to as a Malpighian layers, and this basal, prickle and granular layers are metabolically active region of the epidermis. Now epidermis is thicker in region of the palms and soles and there is absence of sebaceous glands, hair follicles in this area, while in other areas there are presence of these glands and hair follicles. And this thick hairless skin of palm and soles are called glabrous skin, while the hairy skin is also known as hirsute skin. So this is the normal histology slide where you can find this is the part of epidermis comprising of presence of squamous cells in multiple layers and this is the part of dermis. So first of all we will see epidermis. So outer one is the stratum corneum layer. Second, beneath this corneum is lucidum, which is generally present only in palm and sole area and absent in other areas of the body. Now third is granular layer. It is comprising of granules, so it is called granular layer, and these are the layers are called spinous layer. And last the layer is called basal layer, which is at the junction of dermis. So let's see what is stratum basale. It is stem cell constantly undergoing mitosis and regenerate other layers. So basically cells are generating from the basal cell, they are going upwards and they are converted into stratum spinosum. So this layer contains post mitotic cells from the stratum basale. It contains keratin fibrils, melanosomes and Langerhans cells. Stratum granulosum, which is flattened polygonal pycnotic cells that contains keratohyaline granules and ordolent bodies. Stratum lucidum, only in palms and soles, and translucent cells filled with the keratin filament. Sorry. Stratum corneum, which is dead non-nucleated cells filled with the keratin filaments. Now, how will you identify all these layers microscopically? So, first layer, outermost layer is stratum corneum. It appears as a pink lamellated layered compact layer of anucleate cells. So there is absence of nuclei in this layer. And how to identify whether this layer is normal or abnormal? So here it is less than 1/3 of thickness of the epidermis. So whole thickness of epidermis we have to judge, and this layer must be less than 1/3 of the total thickness of epidermis. The characteristic basket wave pattern is due to formalin induced change, and which is called orthokeratosis. Second is stratum granulosum. So it is made up of 1 to 3 layers of diamond shaped horizontally arranged cells with dark purple granules. So here it is 1 to 3 layers only. Either it is increase in some of the condition, or it may be absent in some of the disease condition of skin. Stratum spinosum. It is made up of 5 to 10 layers of polygonal cells with spike-like processes and elongated nucleus. So prominent spike-like intracellular connection is there. So it is called spinous layer. So stratum spinosum because of having spinous processes. Stratum basale, which is columnar cells lying side by side with elongated nuclei, and lucidum, which is amorphous pink layer beneath the stratum corneum and present only on palms and soles. So let's see one by one all layers in detail. So here in this section you can identify stratum basale, which is present at the junction of epidermis and dermis. So here it is this layer above the basement membrane. This is dermis and this is stratum basale. It is stem cell region, so it generates keratinocytes, and it consists of cuboidal to columnar cells, and these cells are having larger nuclei compared to cytoplasm and prominent nucleoli. So it gives marked basophilic appearance to this layer. Stratum spinosum. The layer just above this basale layer is the stratum spinosum. So here in this section you can identify this greenish appearance of this layer here, and in this section these are the stratum spinosum area. So these are 4 to 6 layered cuboidal or slightly flattened cells. They are tapered at the point of attachment to the adjacent cells via desmosomes, and this cytoplasm contains keratin filaments, and this is circumscribing the nucleus and form attachment to the desmosome. So they are attached with another cell with the help of desmosome. Now stratum granulosum. As the name suggests, granulosum means it contains granularity. So during this upward migration of this spinosous layer cells, this granular layer of these cells become flattened and polygonal, and it forms 2 to 3 layers of pycnotic cells. So it is flattened but it contains granules. So here in this section you can identify this granular basophilic appearance of this layer is stratum granulosum. It gives basophilia because of having blue colored granules in the cytoplasm. So here this is stratum granulosum or granular layer. Now what are the components of this granular layer? So one is keratohyaline granules, which is dense basophilic structure in the cytoplasm, and it also contains odd-land bodies, which are lamellar secretory organelles that are also found in type 2 pneumocytes. And these bodies release hydropic glycophospholipids, and this will help to function of selective permeability of the skin, and it makes skin waterproof. Now, as the cellular organelle break down, the keratin tonofilaments interact with the keratohyaline granules to produce keratin, and this keratin is usually soft keratin of the skin, while in hair and nail it is hard keratin. Now, stratum lucidum. This is generally present in the palm and sole, and so it is called glabrous skin. It is translucent layer of the skin above the stratum granulosum. So, here granulosum layer is below this green line, and this green line suggests stratum lucidum. It is filled with keratin filaments and are devoid of nuclei and organelles. So, this is upper layer. Above the granular layer, it is stratum lucidum. Stratum corneum. Corneum means keratinous material. So, stratum corneum is composed of dead non-nucleated cells with soft keratin filaments, and they are known as squamous because of its flattened appearance. So, this is stratum corneum. Now, what are the other cells in the epidermis? One is melanocytes. They are found here in the basal layer, and it is present or it is found for every 10 to 14 basal cells. So, this is melanocytes. Second cell is Langerhans cells. So, it is dendritic cells having dendritic processes. So, it is called epidermal macrophages, and these cells are present in the stratum spinosum. So, here you can identify Langerhans cells. Third is Merkel cells. So, Merkel cells are derived from neural crest and are present in the basal layer, and it is in the close apposition to the nerve ending, and this is basement membrane, which is a thin pink line at the dermoepidermal junction. If we see under HNA stain, and it is if thickened, we can easily identify. So, let us see histology again. So, this is stratum corneum, which is cornified or keratinous layer. This is granular layer. If skin of palm and sole, this is stratum lucidum. This is nucleated cells. It is spinous layer, and last layer of the epidermis is basal layer, and here melanocytes are seen, and collectively spinous and basal layer. So, this spinous and basal layer is also called stratum malpighi. This terminology we are going to use in some of the description of skin lesions. So, you have to remember what is stratum malpighi. Now, along with this epidermal layer, skin is also having dermis and adnexal unit. So, adnexal unit comprising of hair follicles, eccrine glands, erector pili muscles, also having sebaceous unit, and dermis comprising of these reticular fibers, and subcutaneous tissue comprising of fat lobules and fibrous tissue. Now, adnexal unit comprising of sebaceous gland, sweat glands. Sweat glands are of two types, apocrine and eccrine. So, apocrine glands are having secretory portion, duct and acrosyringium, and these apocrine glands are mostly seen in the axillary region, while eccrine unit, it is pale to clear cells, and mostly it is present in the palm and soles. Hair follicles are comprising of, this is the bulb of hair follicle, this is the stem part of hair follicle, and this is called infundibulum, where it connect with the skin epidermis, and this is isthmus part. And if we see microscopically, it comprising of this matrical, supramatrical and matrical cells. This is outer root sheath, inner root sheath, and dendritic and melanocytes. Now, what is intercellular bridges? Because this spinosum or stratum spinosum layer, the cell is, this layer is compact and they are attached with each other with the help of tight junctions. So, intercellular bridges are fine cytoplasmic connection between adjacent keratinocytes. So, this is joining epidermal cells, and it is present only prominently in the stratified squamous lining and mainly in the stratum spinosum. Here it composed of desmosomes, and these desmosomes are made up of desmoglein and desmocollin, and some tonofilaments anchor into desmosomes. So, importance, if we see, what is the role of this junction? Basically, cell to cell attachment having tight junction, adherence junction, these desmosomes, and gap junction. Now, tight junction, it prevents paracellular movement of solutes, and it comprising of claudins and occludins. Second is adherence junction. So, adherence junction, it is having E-cadherin and actin filament. So, this cadherin means calcium dependent adhesion protein. So, whenever there is loss of E-cadherin in any epithelium, there will be metastasis. And desmosome, so it is structural support via keratin filaments, and when these desmosomes are damaged due to autoantibodies, so lesion is called pemphigus vulgaris. So, important note, antibodies again desmosomes are there will lead to suprabasal uh, this lesion, and it is pemphigus vulgaris. So, suprabasal, which is pemphigus vulgaris, which is here intercellular desmosome damage, and hemidesmosome. Here hemidesmosome, it is connecting the cell to basal cell membrane. So, here autoantibodies, if they is present, it is the lesion called bullous pemphigoid. So, bullous pemphigoid lesion is suprabasal, and here it is at the junction of epidermis and dermis. Another gap junction is connexon with central channel, and it is for electrical and chemical communication between cells. And integrins. So, integrins, it is membrane protein that maintain integrity of basolateral membrane by binding to collagen and laminin in basement membrane. So, whenever there is damage to any of these junction, it will lead to some pathology to epithelium, and some lesions are associated with particular damage to this junction. So, it is important to remember all this junction unit. Now, microscopically, these intercellular bridges are seen in between two cells. So, it gives a distinct cell border to particular spinous cell, and this intercellular space appears widened due to tissue processing, and it gives spiny or prickly appearance. So, this is called intercellular bridge appearance. Now, let's see about dermis. So, dermis is comprising of mainly cells are fibrocytes, dermal dendrocytes, histiocytes, Langerhans cells and mast cells, collagen, elastic fibers, ground substance, Adnexal structures like follicular sebaceous, apocrine units, acrine units, nerves, blood vessels, lymph vessels and smooth muscles. So, we have discussed this portion epidermis. Now, let's discuss the dermis. So, dermis is a irregularly arranged region which is filled mostly with the connective tissue. It lies deep to the basement membrane of the stratum basale, and 85% of collagen, which is type 1 collagen and type 3 collagen, are making this dermis. And in addition to this collagen, elastic fibers are there which is helping for recoiling of the skin. Type 4 collagen is located at the dermo-epidermal junction and it envelops Schwann cells as well as vascular endothelium. So, type 1, type 3 and type 4 collagen along with the elastic fibers. Now, this dermis also contains two general types of cells. Like one is permanent cells, which is comprising of erector pili muscles, vessels and nerves, and migratory cells, which are lymphocytes and other leukocytes and related to immune functions. Now there are two layers of dermis. One is papillary layer. So papillary layer name is given because it is having papillary like projections or finger like projection. So this is, here you can see this is epidermis. So it is called epidermal ridges. So these are the downward projections of epidermis called epidermis, epidermal ridges, and these are the layer of dermis which is uh, inside these epidermal ridges. It is called papillary layer or dermal papilla. These are the finger like projections. So these are dermal papilla here and this here suggests epidermal ridges. Now these dermal papilla are less abundant and smaller in thin skin where it is having minimal mechanical stress. And if thick skins are like palm and sole, if you see, it will have a large number of dermapapilla. And type 3 collagen is typically found in this layer. Now second layer. So this is, first layer is papillary layer. Now second layer, this is highlighted with this green area. It is reticular layer of the dermis. And there is generally no clear demarcation between these two areas, but it comprising of reticular cell, reticular fibers, mainly type 1 and type 1 collagen and elastic fibers. Now, beneath the dermis, there is hypodermis. So, here it is highlighted with this green part. So, the dermis rests on this loose connective tissue which is known as hypodermis. It is a superficial facial sheath which intersperses adipose tissue which is called panniculus adiposus. And this fascia reduces friction between the dermis and deep musculature while the adipose tissue is participates in the thermoregulatory mechanism. And there are some small bundles of smooth muscles are also present which is called erector pili muscles that serves to connect the deep part of hair follicle with superficial dermis originates in this layer. Now for the interpretation of skin biopsy, we must know regional variation of skin and its potential diagnostic pitfalls. So if we see or if we get biopsy from back and trunk, normally thick reticular dermis is present, and we must see about morphea and scleroderma, which also give look like this thick reticular dermis. So this is the diagnostic pitfall in case of back and trunk. Now acral skin, which means palm and scole. So here there is thick stratum corneum and there is also a stratum lucidum. So we may confuse it with lichen simplex chronicus. Mucosal skin, which is comprising of diminished stratum corneum and granulosum, pale glycogenated cytoplasm and highly vascularized area. So we must know these are the diagnostic pitfalls of this mucosal biopsy. Eyelid having thin epidermis and basaloid follicular buds. Nose having prominent sebaceous glands. So avoid sebaceous hyperplasia term in biopsy from nose. Axilla contains apocrine glands and varicoid epidermis. Nipple areola having numerous smooth muscle fibers and lactiferous ducts. Foreskin and scrotum having numerous smooth muscle fibers, thin walled vessels, and skin from leg having relatively thick basement membrane in the superficial dermal microvessels. So these are important variation in the skin biopsy uh, interpretation. Now, what are the different types of biopsy we are getting for different type of skin lesions? So one is shave biopsy. So shave biopsy is comprising of epidermis and part of the dermis. So this is called shave biopsy. Second is punch biopsy. So punch biopsy mainly comprising of epidermis and both the layer of dermis, means papillary and deep dermal or reticular dermis. And incisional biopsy. Incisional biopsy comprising of all three layers, epidermis, dermis and hypodermis or subcutaneous tissue. So for the uh, different types of lesion like eczema or psoriasis, lichenoid and blistering lesions are require this type of biopsy, means shave biopsy will help in diagnosis, and mainly these lesions are affecting uh, epidermis and superficial part of dermis. While urticaria, some vasculitis, connective tissue disease or granulomatous conditions where deep dermis is also involved. So in this lesion we require punch biopsy, and in case of paniculitis or some medium and large vasculitis we also require subcutaneous area. So in that diseases we require incisional biopsy. Now whenever we get skin biopsy, if it is unfixed fresh specimen, we can use it for direct immunofluorescence, immunoperoxidase, monoclonal antibody study, some enzyme studies to see mucin, and in case of patient having gout or amyloidosis. And what are the different fixative and stains we can use? So generally, normally always we are using 10% buffer formalin to avoid artifacts like formation pigment and crystal formation. Fixation time must be 1 to 2 hours per millimeter of thickness of specimen. For the electron microscopy, glutaraldehyde is the fixative, and for the direct immunofluorescence, this Michel solution is required. And if you don't have this solution, saline is enough, but we have to transport it immediately to the laboratory. And for the fixation, at least 8 times of volume of the sample is required, and so 10% buffer formalin is measured according to the volume of or the size of the biopsy. And routinely we are using stain for skin biopsy interpretation are: one is hematoxylin and eosin, second PAS stain, and third is Giemsa stain. There are some different stains which we can use in skin biopsy, and these stains are also we can use in other biopsies. So here HNE stain, PAS stain, Giemsa stain, these are routinely used stains. But if we want to see melanin, we can use Mason Fontana, some toluidine blue cells for mast cell granules, von Cosa for calcium, amyloidosis, so Congo red is useful along with this crystal violet, thioflavine, and oil red O for fat, Sudan black for this fat as well as triglyceride and cholesterol, and meson trichome for muscle and collagen. So these are the different stains we can use. And some special stains we can use routinely in skin to uh, diagnose some lepra. It is Phyte Farraco stain. GMS or Grocott-Gomery silver stain is for fungi. Vardin starry stain for spirochetes, and hemosiderin pigment pulse and pulse stain is helpful, and acid fast bacilli are seen with the help of Zell and Nelsel stain. So now we have seen what are the dermatology terminologies we can use to describe lesion, what is normal histology, and how you identify different layers of epidermis, dermis, and hypodermis, and what are the different types of biopsy, and what are the different types of stain. So now what? This is the basic understanding of skin. Now we are getting biopsy. So before we are doing microscopy, we must know some definitions in dermatopathology to describe particular change in skin biopsy. So in next topic we will discuss these all skin microscopy related definitions in detail. So if you haven't yet subscribed this channel, please subscribe and like, share for this important topic of skin biopsy interpretation.